Scientists at Temple University's Lewis Katz School of Medicine have identified a novel gene editing strategy aimed at eliminating HIV-1 infection with no adverse effects on cell mortality. So what is this new approach and how does it work? Welcome to Clinicians Roundtable on ReachMD. I'm your host, Dr. Charles Turck, and joining me today to discuss his team's research is Dr. Kamel Khalili, who's the Laura H. Carnell Professor in the Department of Neuroscience and Chair of the Department of Microbiology, Immunology, and Inflammation. Dr. Khalili, thanks for joining us today.
My pleasure. Thank you very much for the invitation.
Well, let's start with some background, Dr. Khalili. The strategy we'll be discussing targets a gene called mannose oligosaccharide glucosidase, or MOGS. So what is MOGS, and why is it important in this case?
MOGS is a cellular protein which produces in almost every cells, and that product of this MOGS gene is a production of the protein which glycosylate cellular genes that needs to be cellular proteins that they need to be glycosylated in order to exert their function in the cells. So we noticed that based on the earlier notion that maybe MOGS can glycosylate HIV protein, envelope protein, that is responsible for interaction of the virus with the host receptor and then a co-receptor, and eventually virus genome enters into the cells and then basically infect the cells. So we thought that if we can use CRISPR technology to alter expression of the MOGS by manipulating the gene responsible for production of the MOGS, we can make the cell basically become a MOGS-free, and as a result, the virus which enters into that cells will not be able to produce outcoming virus with potential for infecting other cells. In other words, we are disabling a infected virus which is coming out from the infected cells to enter other cells. And it seems that the strategy which we thought it's working in the cell culture, and the elimination of the MOGS results in the production of an outgoing virus from the cells, non-infectious.
And I was wondering if you would talk a little bit more about how that CRISPR gene editing strategy alters MOGS.
Basically, there's two ways we use to inactivate MOGS gene. One was that you can basically stop completely production of the MOGS by manipulating the nucleotide sequence within the MOGS gene, which is responsible for the production of the MOGS. And that's one way. And the other way is that we excise segments of the MOGS gene from the host chromosome. And as a result, cell is making a truncated portion of the MOGS protein, which is inactive. The outcome of both approach is lack of production of the MOGS in the cells. So once the MOGS is not produced in the cells, any virus which is replicating in that particular cell lines or cells become non-infectious. So that is the way you can prevent spread of the virus to the other cells.
For those just tuning in, you're listening to Clinicians Roundtable on ReachMD. I'm Dr. Charles Turck, and I'm speaking with Dr. Kamel Khalili about a novel gene editing strategy for HIV. Now, Dr. Khalili, does this discovery get us closer to a cure for HIV?
Yes, takes us one step closer. We are already have developed a strategy for elimination of the virus from the infected cells by gene editing strategy. And that story is in the clinical trials right now. But we are also developing this additional strategy, basically second generations of the cure of HIV using elimination of the virus and also prevention of the spread of the virus. And for prevention of the spread of the virus, we're using a monk's gene for editing by CRISPR.
And at the research level, I was wondering what the next steps are that you're taking. I think you got into it a little bit, but I thought I'd ask for a little bit more detail there.
For us, the next step is implementing the same strategy in preclinical stage. It means that preclinical could be a small animal model or large animal model. And the model for the HIV in the small animal is a mouse model, which is humanized. It means that circulating in the body of the mouse is a human cell, which is infected with HIV. So these are immunosuppressed mice, which instead of the mouse cellular, the blood cells, it does have human cells, and those human cells are infected with HIV. In the large animal, we are using non-human primate that were infected with the SIV. SIV is a cousin of HIV, which infects monkeys, rhesus monkey. So in order to get into the clinical trial with the combination therapy of the monks and the HIV, we need to perform some preclinical studies for showing the safety and efficacy of that. So that's what we would like to do that as a next step. Take these cell culture studies to preclinical levels, and hopefully based on the result outcome of that preclinical studies, we might be able to enter into the clinical stage for the clinical trials.
Now, before we close, are there any final thoughts you'd like to share with our audience today?
HIV has been with us for more than 40 years, and there has been a lot of information about the virus and how virus interacting with the host, and how virus is leading to the disease progression. And current therapy, which includes antiviral therapy, or ART, has done a terrific job, but has not been able to eliminate the virus. So every time that the patients stop taking the antiretrovirus, the pre-existing virus in the body rebounds, reactivated, and then put the patient individual at risk for the development of the disease. Our strategy is to eliminate the virus from the host genome. And by doing that one, the individual no need to take any antiretroviral drugs for HIV, and then permanently cure the disease by eliminating your virus. One thing that I didn't mention is important to note that when the cells become infected with HIV, viral genome incorporate in the host gene, and they remain at the silent stage until it replicates, which frequently it replicates, and result of that is killing the human immune cell, T cells and the myeloid cells. So we look at the virus at that stage, the face of the virus and host changes. Virus become like a genetic disease. The disease that once it was infectious disease become like a genetic disease because of the viral genome is now incorporated in the host gene. So in order to permanently eliminate the virus from the cells, we must use genetic approach and gene therapy, and then gene editing, which includes use of CRISPR, provide the excellent tool to achieve that goal. And I think that we are entering into time after 40 years that we learned that the gene editing can serve as a potential tool for elimination of the infectious diseases, including HIV, by manipulating the structural organization of the viral genome and making them become inactive.
Well, this has been such an interesting look at some exciting research aimed at the elimination of HIV infection. And I want to thank you, Dr. Khalili, for joining me today and for sharing your team's research with us.
Many thanks. Thank you very much.
For ReachMD, I'm Dr. Charles Turck. To access this and other episodes in our series, visit reachmd.com/cliniciansroundtable, where you can be part of the knowledge. Thanks for listening.